Welcome to BB 101 course at IIT Bombay. Professor Srivastava is going to deliver his lecture in Molecular and Cellular Biology module shortly. Please mute your microphone and be ready with pen and notepad to take notes. Thank you. Welcome to BB 101 course at IIT Bombay. Please mute your microphone and be ready with pen and notepad to take notes. Thank you.
Hello and welcome to Lecture 6 of BB 101 module. In the previous lecture, we learned about the foundation of genetics. As you would remember, Mendelian genetics always assumed the existence of genes or factors, as they were called then. There was no physical evidence that such a thing existed. It was an abstraction. But soon, scientists found the evidence of those factors. Today, Professor Sanjeeva will tell us about that evidence. Mendel's proposed hereditary factors during sexual life cycles. Today, these factors are called chromosomes and genes that are both present in pairs in diploid cells. Homologous chromosomes separate and alleles segregate during the process of meiosis. The fertilization restores the paired condition for both chromosomes and genes. From the first solid evidence associating a gene with a specific chromosome, from the work of Thomas Hunt Morgan in the 20th century, to experiments performed by Alfred Hershey and Martha Chase in 1952 showing that DNA is the genetic material, we will learn how slowly and gradually, but surely, the chromosomal and molecular basis of inheritance was established. While it may seem all too obvious today, this journey is surely a masterclass of human ingenuity at its best. Let us join Professor Sanjeeva and learn about chromosomal and molecular basis of inheritance in Lecture 6 of BB 101 module. Thank you.
Hello students, welcome back to BB 101 course. In the last class, we talked about Mendelian genetics, and you learned that Mendel performed some very elegant experiments on garden pea, which resulted into fundamental principles of law of segregation and law of independent assortment. We also learned about various examples linked to Mendelian genetics, the experiments on blood grouping, as well as how the law of probability can be utilized along with Punnett square to derive the possible offsprings in F1 and F2 generations. Continuing on the same theme, today we are going to look at another model organism, from peas to Drosophila. The fruit fly Drosophila melanogaster is one of the experimental model which has been heavily used by genetics scientists who are working on crossing, because it is very small fruit fly which you might have seen on the peel of a banana, and that can be grown easily in the lab cultures with banana peel, and they have many strains of that. One could also introduce the genetic changes and try to maintain different strains. Genetic crossing can be very fast because the flies are going to grow very quickly. So therefore, Drosophila melanogaster is one of the very good model organism to understand genetics principle. In this slide, another scientist, Morgan, he performed the experiments. On one hand, he tried to understand that what Mendel has reported, whether those facts hold true even on the not from the plant, but also on the Drosophila. At the same time, also that Mendel's law did not have any detail regarding where these genes are located, where are the chromosomes, because there was no genetic basis to these rules. So Morgan actually went one step further, and he tried to convey that how not only that Mendel cross and those laws are still applicable, but also there is a genetic basis to these involvement, and there are genes linked on the X or the Y chromosomes, the sex-linked inheritance which happens, and where the genes are located on the chromosome. These evidences came from Morgan's experiment. So today in lecture 6, we will talk about two topics, chromosomal basis of inheritance and molecular basis of inheritance. In the chromosomal basis of inheritance, we will start first with Morgan's experiment on Drosophila. And those experiments, we will see how interestingly and elegantly Morgan showed that Mendel's experiment on pea plant could be performed on the fruit fly, but also gives the idea about the genetic basis, where these genes and genes are located on the chromosomes. We will then talk about sex-linked inheritance, and I'll show you different crosses, how different idea for the genes location can be very useful for us to understand about the possible pedigree of the family tree. We'll then talk about the concepts of genetic recombination and linkage. And again, some elegant experiment done by Morgan will be very useful. Finally, in this topic, we'll talk about chromosomal abnormalities, because there are many syndromes, many disease happens because of abnormalities in chromosomes, and you will get some idea from this topic. We'll then talk short topic on the molecular basis of inheritance. We are going to show you some classical experiments which were done in 19th century, and those are very elegant experiments which showed beautifully what are the fundamental concepts of different molecular biology. For instance, Griffith, he showed that which are the transformation factors. He did those experiments. Hershey and Chase, these scientists tried to prove that whether DNA is a genetic material or protein is a genetic material, and those experiments were performed. And then how DNA replication happens. We learned about DNA replication happens in the S phase when we talked about cell cycle regulation. So Michelson and Stoll, these scientists, they worked on the DNA replication, and while there were other scientists who also performed different experiments, their theory on semi-conservative replication got more prominence. So we have two different topics to cover today, and it will be a good idea for you to take your notepad and the pen, and it's good in the genetics. Whenever we are going to do any cross, please do it yourself. You will find it very interesting and very easy if you start doing it yourself, right? So let me now move from my office directly to the classroom of BB 101 of previous year and take you there to discuss about chromosomal basis of inheritance. I must say that, Mendel made huge contributions for identifying that there are certain factors, heritable factors, which transmit from one to next generation. But, quite long time people did not know that what those factors are. And then, also people did not have too much information about whether those factors, in which manner they are inherited. So, few scientists started making more investigation. And one scientist who had made huge contribution in the area is Morgan, and I'll talk about some of his experiments. But before, what we are going to talk is, are the genes for the P property which we are talking, are they present on the chromosomes, and do they segregate as a part of the divisions, meiotic divisions, the way we have been studying the meiosis process, right? So in this case here we have yellow and round, and we have green and wrinkled seeds. As a part of meiosis, these gametes are separated, and if these gametes contain the genes, those genes which are giving the properties like yellow and round seed or green or wrinkled seed, so are they also getting segregated as a part of these divisions which are happening. So genes are located on the chromosomes, and what is the behavior of these chromosomes during the meiosis process, and whether Mendel's law, which we have talked about the segregation and independent assortment, is also valid as a part of these chromosomal basis of separation. Little complex slide, but one could start thinking and refreshing yourself about meiotic cell division. So as a part of law of segregation, the two alleles for each of these genes will separate during the gamete formation, which you can follow the same what we have done in the meiotic process. And the law of independent assortment, it means alleles of genes on the non-homologous chromosomes would assort independently during the gamete formation. So it's very well color coded here in the red and blue, showing that your capital R and small r alleles for the same gene properties, how in the meiotic process they are getting segregated. So these are the kind of assumptions made, thinking about whether what Mendel has discussed, whether that has any basis at the chromosomal level. So far what we talked is essentially that Mendel's dihybrid crosses one could correlate also at looking at the behavior of the chromosomes during meiosis. During meiosis, these homologous chromosomes, they get segregated, and therefore alleles also get segregated. The behavior of chromosomes in meiosis process of the F1 generation as well as after the fertilization in the F2 generation could be observed by what Mendel has already shown in his crosses. So while these are some assumption, but there is no experimental evidence that how one could prove this. And this is an area where I must say that another scientist who made huge contribution is Dr. Morgan, who studied the Mendelian inheritance and what are its physical basis at the chromosomal level. So this is a scientist for whom we are talking, Thomas Hunt Morgan, Columbia University, and he selected a model system which is Drosophila, or the fruit fly. So far we have been talking mainly about pea plants, and but at some point I had mentioned to you that many people like to work on Drosophila because again it comes in many properties. You can grow them very fast. I'm sure you would have seen Drosophila in some time on the banana peel. You will see those small flies with red eyes. And people make these cultures of these particular Drosophila stalks they make. This is showing you here lot of labs which works in the genetics and developmental biology area. They have the stalks of these Drosophila strains, and these Drosophila you can make mutations in different genes, and you can keep those and keep growing those, which can be very handy for doing lot of experiments and testing your hypothesis. So what is shown here, you have small tubes, and in those tubes you are having banana peels and some nutrients on which these flies can grow, and people store them and mate them and make sure that they are maintained well for doing a lot of genetic experiments. So this is where we are right now, but at that point in 19th century, Morgan started working on Drosophila and thought about looking at Mendelian genetics at the fly level. So let's see what kind of experiment he did. Finally, he provided evidences that chromosomes are indeed the location of Mendel's heritable factors. So I have there is no suspense on that. I already made the conclusion, but now let's see the experimental system and what kind of things he did. So now nomenclature of the things, the way we have been discussing from the pea plant will slightly change. Here we are talking about phenotypes of Drosophila. Some characteristics, if these are commonly found, for example red eyes, that is wild type character, and if a deviation of that is found, that is known as mutant type behavior. So if this particular eye is white eye, which is not commonly found in Drosophila, that is a mutant type behavior, whereas the red eye is commonly found normal phenotype, that is wild type. So I'm sure now you are familiar with the wild type and the mutant terminologies. Let's look at some of the how you can denote them. So far we are talking about in the pea plant context only simple nomenclatures. Now let's add a little bit complexity here. When we say wild type, you are denoting with a plus sign, and when you have a mutant, then there is a no plus sign. We are also showing here male and female chromosomes. So this is a male Drosophila, so XY chromosomes, and X is having W suffix on that or superscript on that, so that is a mutant behavior. Now this particular thing is XX, a female, both contain W+, that is a wild type, which will give the red eye pattern. Keep this in mind. I think there are some crosses which we have to do. which now we have to use these kind of nomenclature. So if in a given cross, some point, we'll talk to you about a given property, its wild type and the mutant behavior, you can denote those with plus or without plus. Alright, so now let's see what kind of cross Morgan did. So he took these wild type female fruit fly as well as mutant white-eyed male fruit fly, and then observed their progenies in the F1 and F2 generation. So in this case, the female is red-eyed here and male is white-eyed in the parent generation. In the F1 generation, all the offsprings, they showed red eye. Just imagine what Mendel's experiment were, right? So purple was the dominant, and now that was shown in all the F1 generation. Something similar he also seen. Now he wanted to follow this particular cross in the F2 generation, and I would ask all of you now to start thinking and start making this kind of cross. So let's start doing that. Alright, so we have the so you have to now start making X and Y chromosomes, and then also you have to denote what is a wild type and what is mutant, right? Then only you can say that offsprings, how many are male and female and what are their characteristics. So let's try out simple, the same way of Punnett square. So let's draw only for F2 generation. So this is Y chromosome, and we have this X chromosome, which is having W plus. All of you are doing that, right? All right, I'll come and see your results, all right? I'm sure right now it's not very difficult for you to do the crosses, but you have to assume that you have to know the terminologies, you have to know how genetic crosses can be done in different type of properties which we will describe. Alright, so I'm sure all of you have done your cross by now. So what is the phenotypic ratio? We are looking at one property, which is red eye versus white eye. White eye is the mutant property and red eye is the wild type property. And these signs are for male and female, which also is important to draw those. So it is not so difficult, right? Because you have done already this kind of crosses for Mendelian genetics in the P, and that time 3:1 were very simply defined. Yes. Alright, so a good observation that whether we have no gene on the Y chromosome, And that is, I think, a right observation, that what Morgan also proved here, that certain genes, they are only carried on a specific type of either X chromosome or Y chromosome. So some properties are linked to specific sex chromosomes. They are not present in both the chromosomes. And therefore, and this observation did not come beforehand, it came only after this cross. So now if you realize, we have only one white-eyed male, right, which is the mutant type. So because he also observed 3:1 ratio, 3 other flies were red-eyed, 2 of them female and 1 male. But one of the white-eyed male, he was observing this only male. So based on that, when he made these crosses, he realized that the segregation is happening, but there is some sort of link to the chromosomal basis of that. Probably these genes are located on some specific chromosomes, X chromosome or Y chromosome, and that's why they are seeing this mutant behavior is coming only in the male type here. So I think it's important in genetics that you should make those observations for a given cross and then use all the properties, label properly, and then probably you cannot make any mistake. So this is kind of cross which we just talked. We have these gametes from the sperm, W plus and the Y chromosome here. From the X we have W plus and W. And now if you are making these crosses, you have only one white-eyed male. You have remaining 3 flies which are all red-eyed. Yes. So phenotype wise there 3 are red-eyed. Phenotype, if you look at of the 4, it's 3:1. Alright, so what we just discussed the cross, the main points of this cross are that the mutant or the white eye trait, that is recessive as compared to the wild type of the red eye trait, which is W plus. In the recessive trait, white eye was expressed only in the male in the F2 generation. I think you had rightly observed this, and that led Morgan to conclude that eye color gene is located on the X chromosome and not on the Y chromosome. So while that segregating, it is some specificity that where these genes are located. So conclusion from the experiment is, we saw the evidence that chromosomes are indeed the locations of what Mendel has defined, those heritable factors. So Mendel was only able to say that there are some factors which are transmitting from one to the next generation. Morgan was also able to show that there are some chromosomal basis to them. Alright, so genotypic ratio, you can say in the female what is the genotypic ratio, in the male what is genotypic ratio, and overall genotypic ratio of all the offsprings. So in that way, yeah, it can be, you can say that among the female this is the genotypic ratio, among the male this is genotypic ratio, among all of the 4 what is the phenotypic ratio, or even you can split into male and female and you can put in the ratio form. So if you think about human and the chromosomal basis of sex in human, we have 44 pair of the 44 autosomes and XY chromosomes are there. So these 22 plus X or 22 plus Y part of the sperm and 22 plus X part of the egg. This is known as the XY system, where X and Y chromosomes and the various genes which are linked to them are getting segregated. So sex of a child actually depends on whether the sperm contains X or the Y chromosome, because egg will always carry X chromosome only. So the sex of child will be derived from the sperm, whether it's coming from the X part or the Y. So there are many sex linked genes which are only found on the X chromosome or the Y chromosomes, and the Y linked genes are actually much smaller in number. Only around 78 genes have been found on the Y chromosomes, whereas on the X chromosome almost 1100 genes are there. So a lot of properties are only uniquely present on the X chromosome or on the Y chromosome, and they will follow that pattern coming from these chromosomes. If you look at this circle, can you read something in that? How many people cannot read? Cannot read? How many people cannot read? Alright, so if in case you are not able to read, I think you should pay some close attention to this and you should go for some test. You might be not able to distinguish the colors so easily, and this is reality, right? Because for many of the exams, especially for the railways, military, etc., they also test for your color blindness. Can you distinguish the colors clearly? So many of the disorders which are sex linked includes red-green color blindness, muscle dystrophy, you have night blindness and hemophilia. So many of these diseases are actually derived based on the transmission of X-linked recessive traits, especially for the red-green color blindness. So let's think about the red-green color blindness, which is much more apparent case. Let's think about again some of the ways to denote the conditions. Please pay attention to this slide. A lot of questions are going to be based on this. So for a color blindness to happen, this gene is going to be on the recessive allele, which we are denoting from the small n. If an individual is unaffected or normal eye, normal vision, then we are showing from the this white square. If it is carrier, we are showing from the light shade. If color blind, from the dark shade. So this case, let's think about female and male. A cross is happening. In this case, it is normal. When we have normal phenotype, it means capital N and capital N, right, on both the chromosomes. And if it is carrying the recessive allele, which is small n, then only you have the disorder. So if I am just showing you this, what is this cross is between what type of male and female? This male is
colorblind, yeah.
And female?
Female is unaffected.
Alright, so female is unaffected and male is colorblind. Alright. Now let's take second situation. In this case, what will happen? What female is?
Carrier.
Yes, and male?
Unaffected.
Male is unaffected and female is carrier. Let's take the third situation.
Female is carrier and male is colorblind.
Alright, so I am hoping that in addition to looking at just the colors, you are also paying attention here that whenever there is a small n, which is recessive allele is there, with the male it is only it has to be only one on the X chromosome, so it's always getting color blind. Now in the female, even if it is present in one copy, then still it is only carrier, it is not showing the full effect, right? Now let's try to take these conditions and then do the crosses and see what will be the fate of these offsprings coming from these crosses. Please start doing that. So that's the first situation we had talked. Second one is that we have male which is normal and female is carrier. Third cross, we have male him colorblind and female is the carrier. All right, all of you are doing it, right? Also should write based on the X, Y, which are male and female. All right, so I'm sure all of you have done the cross as well. So you have to now do these crosses and give me the answers for each of the cross. What will be the progenies look like? And when the questions will be asked, you will be actually given a situation when that if the cross happens and the kind of child which are born, 50% of them are carrier and 50% of them are normal. What could be the genotype of the parents? So then you have to go backwards. So let's go one at a time. The first cross, is everybody trying now? Everybody is comfortable? How about this side? Okay, so let's think about different crosses which we have done. The first situation is when we have a color blind male and we have the normal female. From this, the progeny which are born, of them 50% of them are carrier. Okay, you will not see apparent any change in the phenotype, but the 50% of the child are actually going to be carrier. So a color blind father is going to transmit these alleles to their daughter. It's only transmitting, and it is not going to be apparent in the next generation. Second situation, the second cross, we have the female which is carrier and now male is normal. In this case, out of the 4 progeny, there is a probability that one of the male child will be color blind and one of the female will be the carrier. Now let's think about the third situation when we have a color blind father and mother is a carrier. In this situation, you have 50% child who are going to be color blind and 25% of them will be carrier. So many times if a family has the history of carrying these kind of disorders, so it's good idea for them to do genetic testing or get genome counseling before marriages, because you want to know that what could be the fate of those small genes and whether they will be apparent in the progeny. And therefore many people make those kind of choices. In this case, if let's imagine here, in this case, even if the father is actually color blind, but as long as mother is totally normal and not carrier, then the progenies will not have any effect. But if this is the third situation is there, the father is color blind and they are not doing any genetic testing, they are marrying with anybody, and then that female is also carrier, then the possibility is that most of the child will be either color blind or they will be carrier. So these are reality, and many times when these genetic disorders happen, people do these genetic testing to ensure that they can avoid this situation. Okay, so these things can be just done in the same kind of mathematical probabilities the way you read many other mathematics questions. So conclusions from these crosses are their fathers pass X-linked alleles to all of their daughters, but none to their sons. And these child daughters, they are actually carriers. They are going to carry from that generation to the next generation. Mothers can pass the X-linked genes and alleles to both the son and daughters. Any male which receives the recessive allele from the mother will express the trait. Sometime I'll just give you this kind of statement and I'll ask you to show this is true or false by doing a cross. the small cross which I showed you are very simple, but it becomes difficult when you have been just given a statement and you have to derive the crosses based on that. So I am hoping that from this exercise you are getting more comfortable now, and you should be very easily able to derive those crosses now. So far more males than females actually have X-linked recessive disorders. So absence of one or more of the proteins which is involved in doing the blood clotting is not found in this particular disease condition, and therefore there is excessive blood loss happens from the wound site. In fact, nowadays, because of all our advancement understanding of doing genetic engineering and protein research, it is now possible that we have those clotting factors which in the purified form present, which could be injected in the individuals, and one could try to overcome this deficiency. But of course that time it was much more serious issue. And what was more interesting, that people started observing Queen's family, Queen Victoria's family, that many of their descendants were actually passing these traits. So many of the royal family children were carrying these alleles, and therefore some of them started showing these symptoms, and then it became more apparent that it's not only one child which is having problem. There are many from that family who are having these issues, because their intermarriages were from the very small group of individuals from Spain, Russia, etc. But this was definitely one of the very interesting fact. It was happening in royal family, so became much more apparent, and everybody started noticing this problem. And that time some scientists published a study on hemophilia, and then they this thing became much more like a case study for all the scientific community to find out what is happening in the royal family and how the genes are getting inherited. Okay, so the conclusion is that you may now overcome some of these deficiencies because we have understanding of how to synthesize these proteins and the clotting factors, which could be injected in the individual, and that may prevent the blood loss. But nevertheless, understanding the genetic basis is very crucial, and these kind of genetic counseling per se can actually help us to avoid many of these kind of mishappenings. So by now you are familiar with the chromosomal basis of inheritance. You now know that Mendelian inheritance has its physical basis in behavior of chromosomes. It is the behavior of chromosomes during meiosis that accounts for Mendel's law of segregation and independent assortment. Morgan independently tested Mendel's experiment and provided evidence that chromosomes are indeed the location of Mendel's heritable factors. You also studied sex-linked inheritance and how Y-linked genes mainly help determine sex, while X-linked genes help in determining genes for many characteristics but unrelated to the sex. Morgan, in addition to making contribution for understanding the chromosomal basis of this inheritance, he was also making some very interesting fundamental observations. If you now think about going back to meiosis, at some point we have said that the genes get recombined, and that's what gives lot of uniqueness among the progenies, right? Among the child you see lot of differences because the genes get recombined. So he was observing this, that whether the genes recombination event happen, or there are some genes which are very closely linked to each other and they are not getting recombined. So the production of offsprings with combination of traits which differs from the parents, whether those are passing together or they are getting segregated. So think about the classical Mendel thing first, and then we come to Morgan. So in Mendelian experiment we have seen these are the parental type offsprings, and then when we did this dihybrid cross, we also saw some recombinant offsprings appeared. Recombinant means some new type of features appeared along with the original combination of the characters. So two non-parental phenotypes were also seen in the offsprings, and these are known as recombinant types or recombinants. So if you remember from the meiosis image, these chromosomes are exchanging a small segment of it at a site which is known as crossing over. So from this part, from the male and female gametes, there are certain interchanges happening, and now you can see in the recombinant A and recombinant small a, this part is actually transferred. So all the genes which are part of this one has now come in this chromosome, and all the genes from this part has come to this chromosome. So this is kind of concept which we have briefly seen in the meiosis part, and Morgan was trying to investigate this in much more detail. So just to refresh you, recombination is an event when these genes are getting recombined and they are getting exchanged from these two chromosomes, and at a site which is known as the crossing over site. Alright, so this part we have talked in the meiosis prophase 1, just to be precise, and we have seen that a set of proteins are getting exchanged as a part of it, which we say as a crossing over event. So how does, if the genes are linked or they are far apart, how does that information can be transmitted from the one to next generation? And Morgan was always using the fruit fly as a model system, and he wanted to study these investigation also in the fruit fly. So let's now increase little bit more information from the fruit fly experiment. And in this case, now we have two different type of fruit fly. One is wild type, which is having the gray body and the normal wings, and second is a double mutant, which is black body and the vestigial wings. Just to simplify this part, if you make these kind of crosses or two properties like this, that if we are saying wild type, it means it is plus, right? And we are showing on both the chromosomes, so B plus and VG plus, B plus VG plus, and we are doing a cross with double mutant. So what is shown here, B plus is it means showing on both the chromosomes, right? So this is how the cross should ideally look like. And so now if you take this situation that you have a wild type with two features, and what you are looking at is studying two genes, and are they linked to each other, or are they going to separate in the process of recombination? Now in the F1 dihybrid cross, all of them are looking like the same, the wild type feature what we had. And now if you want to do a test cross from this, think about Mendelian test cross. So what would you do the cross now? If you want to do test cross, F1 whatever is the phenotype shown here, this F1 you want to do a test cross. So what will be the genotype of the other fruit fly? You want to do a cross of this particular fruit fly, and you want to do a test cross. So what we did for a test cross in Mendel's PE experiment? Anybody recall? Homozygous recessive. Is this homozygous recessive? This is right. All right. Well, so now can we see this thing? This wild type of F1, and we do a test cross with homozygous recessive double mutant. Please do that. Write on these gametes and make the square. So now you can write these gametes. So how this will look? Okay. Now you try other.
You are trying to do the test cross. So what does it mean? Like you are saying that there are 4 different types of eggs which will produce, which you will cross with the test cross.
Let's discuss after doing the cross first. All right, and now if you can read from here, these are gray body and normal wing, and this is black body and vestigial wings. Could you also write down what will be the phenotype of these flies? Write down what are the phenotype of these flies. Plus means wild type, and
when we get B plus,
yes.
then we consider that B plus is dominant over B.
So that's why I was telling that in all of these problems it's a good idea to write B plus, VG plus, and B and VG, because these are like this, because these are on different chromosomes. So it's ideally same thing. But when it is plus here, it is mean it is wild type, and if it is not plus, it is mutant. So we have now these 4 different type of phenotypes, and what we are observing here that one is gray normal, one is black normal, one is black vestigial, and one is gray vestigial. So in addition to the 2 which were original phenotypes what we had, we have 2 different new phenotypes as well. So what Morgan was trying to understand from the experiment, whether these genes are very closely linked to each other or they are very much far apart. So if you think about, just imagine that on this chromosome, if this part is getting recombined, and these 2 genes which we are studying, one is this part and one is going to get separated, it means these are not linked. But if these 2 genes are here or these 2 genes are here, these are going to be linked, right? If they are very close to each other. So then if they are close to each other, so even if they are getting recombined, still they are transmitting together. So therefore these 2 genes, a higher proportion of this looks like they are linked to each other. But there are some progenies, some flies which are also found of other phenotype, which may be that some recombination event might have happened. So he was the first time he was looking at not only that how genes are being transmitted, but where they are located on the chromosomes and are they linked to each other or not. So this is where he was looking at the linkage phenomenon, and what he concluded that most offsprings had parental phenotype, whereas very small number, relatively very small number of the flies were also having the non-parental phenotype, which means that there might be some mechanism which breaks this particular type of linkage, and some recombination events might happen which is being transmitted from one to next generation, which happens usually during the meiosis process. Alright, let's take a short break and let me show you a clip. Alright, so it's not going to be a quiz question, but what is this movie? Very good. And what is the character? What is this character? Character name? Oro. And what is this disease come from? From which syndrome? That will be a question. Alright, so I'm sure you would have appreciated when you would have at least watched the trailer of this movie that there are some child who might be suffering from these kind of chromosomal glitch to say, and those sometimes could just be because of one gene defect, and in this case it was based on a lamin A gene which is defective. So there are many diseases which happen because of certain alterations happening at the chromosome level, and let's kind of not discuss some of these kind of genetic disorders which are happening because of abnormality in the chromosome number or the structure. So when we want to study about all the chromosomes, if you think about at some part of developmental study we talked that you want to map the karyotypes, you want to look at all the chromosome patterns, and it's just color coded differently for each pair of chromosomes. So it is possible that while various recombination events are happening, various genetic events are happening, some part of chromosomes get structurally changed, and it don't happen always, and don't happen very often, but once in a while it happens, and those may happen that in a small part of the chromosome get deleted, and in this case this D part is deleted. So this is known as deletion of the chromosome. So all the genes which are a part of that part of the chromosome is going to be absent, or it can be inverted. For example, if the gene sequence was A, B, C, D, E, now B, C, D became inverted, so now you have A, D, C, B. So many of the the cascade of the gene signaling will get changed because of this inversion part. Or a segment of it, like BC in this case here, gets duplicated. So now you have 2 copies of B and 2 copies of C. One more situation is translocation. A part of these chromosomes move from one to other chromosome, like AB in this case moved to MNO and MNO moved to AB. So now they became very different characteristics. Or sometimes it can be non-reciprocal. It means it is just going to get attached to other chromosome, not the vice versa. So there are many things which can happen at the chromosomal structural level, and these are all different type of aberrations which result into different type of diseases, different disorders. For example, you see this child here suffering from Down syndrome. One of the very commonly found syndrome, which is having 3 copies of this 21st chromosome, which is known as trisomy. Trisomy means 3 copies of this chromosome. So Down syndrome is usually result of an extra 21st chromosome. So now their total chromosome number will be 47. So there is a disbalance, and these trisomy of 21 is now people do test for this at the prenatal level for testing whether the fetus might be carrying this syndrome or not. So another syndrome which is something very similar, which is trisomy of 18 chromosome, that is known as Edwards syndrome. You can see this particular phenotype here, Turner syndrome, which is based on absence of the certain genes from the X chromosomes. And now here you can see a phenotype for Patau syndrome, which is a trisomy of chromosome 13. So 3 copies of chromosome 13 is present in this case. So many of these things are some abnormalities which happen because of the way chromosome would have shown some changes at the structural level. And we have seen this example that even sometimes a specific gene like lamin A gene in this case having some mutation, and as a result it resulted into this Hutchinson-Gilford progeria syndrome, and then this kind of child is being born, which now people can do genetic testing and try to find out these kind of genetic abnormalities beforehand, and that's where our understanding of molecular biology and lot of genetic tools are becoming really important in medical field. But what happens at the plant level? So many times plants also show these kind of changes, but not always these changes are very detrimental for plants. So in plants what is more common occurrence is that certain chromosomes just totally get duplicated, or they make 4 copies or 6 copies or 8 copies, and this phenomena is known as polyploidy. You have multiple ploidy level of those chromosomes. So rather than only one chromosome making extra 3rd copy or 4th copy, the entire chromosome set has become duplicated. And this is what is shown you here, that 2n, 4n or the 6n of the whole chromosome sets. Look at this particular watermelon. I'm sure you would love to eat watermelon without seeds, right? Seedless watermelon. And those are actually polyploids. Those are having extra set of chromosomes, and sometimes those result into some beneficial properties in plants. For example, many of the high yielding wheat variety, those are hexaploid wheats. So it's not always that if you have some extra chromosome, those are going to have problems. In this case it is much more advantageous. But what is important here to note, in plants we have full set of chromosomes getting duplicated, and that's usually is much more easy to retain for the cell as compared to only one chromosome getting extra. So that's where I think you can see these are still giving us some positive results in the plants. So we have been discussing about Mendelian genetics, and we have seen many examples in which way understanding the genetic laws can really help us to do a lot of genetic testing. In the same light, I think it's important for us to learn how new modern biotechnology tools have started making huge difference in day-to-day medical applications. You have just seen one video where you may have realized that in which way personalized medicine is not any more a fiction. If you understand an individual's genome variation, what kind of mutation might be happening in this particular individual, so then you can give some medicine which are very much could be recognized by the receptors of those mutations, and then it will be much more effective for that individual as compared to you are just giving some generic drugs to every individual. And therefore many times for a given treatment you may realize that you have a lot of side effects, and those side effects are just result of us not knowing this exact way of modality of the treatment. So in molecular basis of inheritance we had discussed about some of the classical experiments, and then I'll talk to you about some more classical experiment in which way some of the fundamentals that what is the genetic material is the DNA or the proteins were established. And similarly many of the transforming principles as well as how DNA replicates, those kind of laws were how they were derived. We are going to discuss about those experiments. Can a genetic trait be transferred in two bacterial strains? There are two strains, one is having capsule with S strain, one is not having capsule R strain. The one which is having S strain may cause pneumonia, other one is not going to cause pneumonia. And this experiment was done by Griffith where he proved the principle of transformation. So we had seen the first condition that if you have S capsule which you are now injecting in the mouse dies because of the virulence from the capsule. If you have living R cell, they are non-pathogenic, they will not have any impact. So mouse remain healthy. If you are having the heat killed S cells, it means you are killing the capsule layer, then that makes it non-pathogenic, and again mouse will remain healthy. If you have last situation like this, when you are adding a mixture of heat killed S cells and you are adding the living R cells, what was the phenotype for the mouse? Mouse died, and that was because of what happened in this experiment, in the last one?
The last one it gets killed because the R cells will develop the S cells traits. It will.
And how that happened? So in which way R cell acquired the S cell characteristics? So in this case probably some material, some DNA from the S cell got transferred to the R cell and made them pathogenic, and this is what resulted into this phenotype which was mouse died. So what Griffith then concluded from the experiment that if we are injecting both non-pathogenic and pathogenic, but there is some kind of transformation is happening, some substance is moving from the S cells to the R cell, and that is giving it the property which is making it much more pathogenic and creating the mouse to die. So this is kind of conclusion from the this experiment. So the living R bacteria got some substance from the heat killed S cells, which is like a transformation is happening in this particular type of bacteria, and therefore this particular phenotype was observed which is not so much common, which was not expected, and that's where he first time realized there might be some material could be transformed, and that resulted into Griffith's transforming principle or the factors which are known as the DNA or the transforming factors. So now let's come to the next question which we started discussing briefly. Is protein or DNA the genetic material? And now who will explain this experiment? So we have been saying, all the genetics based on DNA, that DNA is going to transfer from one to next generation, right? But why by default we assume that DNA is the only thing which is going to have those properties which is going to get transmitted from one to next generation? It might be even protein may have the same property. So somebody would have done it first time and thought about this type of experiment, right? And these are the two scientists, Hershey and Chase. If you remember, we discussed briefly that one could use some sort of labels to find out or to track these kind of molecules. In 1930s, 50s, that time, most of the experiments were done very elegantly just by using some very basic reagents and the labeling strategies and looking at things in the radioactivity. So if you know that you can label DNA because you have the phosphorus backbone in the DNA with the 32 phosphorus, you can label DNA, or you can label protein with some methionine residues are there in amino acids. With the sulfur you can label proteins. So they use that property, and let's kind of look briefly. So just imagine they used one of the bacterial strain, and now there is a virus which is phage. A phage which is it can eat bacteria, that is bacteriophage. So now it is this particular virus is there. So virus for its propagation transfers some genetic material to the bacteria, and therefore then it can have its multiple property, it can propagate in bacteria and make multiple copies of its own, right? So what they are thinking, can we label both the DNA of it, if there is some DNA component of virus, or even the protein? If we are labeling both of them, and now let's see what is going inside the bacteria, which is going to create multiplication. So that will be probably one which is going to have all the genetic information, so that one could then extrapolate information from it whether the DNA or the protein have the genetic information. So to do this experiment, they use a very simple technique, analytical instrument, which is known as centrifuge. In centrifuge, I'm sure you have seen washing machines. So it's something like washing machine, where you are using a very high centrifugal force, and let's say you have these are the rotors in which you are keeping the tubes. So let's say one of the tube is kept here, and when you are using very high centrifugal speed here, let's say 10,000 RPM, etc. So then based on this particular speed, if you have this tube here, whatever the bacterial membrane and the contents are there, they will just come as a part of the bottom part, which will be pellet, and anything which is liquid, clear part, that will remain as supernatant. So if whatever is going inside the bacteria along with bacterial membrane and debris, it will come inside, which will become part of the pellet, and anything which remains out will become part of the supernatant. That was his assumption, and he had labeled both protein and DNA. So he was trying to see from radioactivity what is going inside the pellet and what is going in the supernatant part. When he did this particular labeling experiment, what he found that in the pellet he could see radioactivity of the DNA. So based on this information, and whereas the protein was found in the supernatant part, so looking at this information, he was able to conclude that the DNA is going inside the bacteria from the phage, whereas the protein remains outside, it is not entering inside the bacteria, and therefore protein he can see in the supernatant part and DNA is coming in the pellet part. Just by doing the simple labeling experiment, he was able to conclude that the phage DNA entered the bacterial cells, but phage proteins did not. So these were some very classical experiment done in 1900 that century, which has resulted in a lot of fundamental information. And if you think about it, I think these are not so difficult thing to do, just need some sort of concepts to be tried out. I'm sure we have studied about chemical composition of DNA. You have been taught looking at 2 DNA strand double helix model of DNA. Watson and Crick, these are the scientists who elucidated the structure of DNA, and they got Nobel Prize for it. Very briefly, you have AT and GC base pairs. Whenever you have A, you will have the T with the 2 hydrogen bonds, and G with the C3 triple hydrogen bonds. So this kind of pairing will be there, complementary base pairing will be there. And now whenever a scientist, actually Erwin Chargaff, he derived the rule that percentage of A base pair will be equal to percentage of T base pairs, whereas percentage of G base pair will be equal to percentage of C base pairs. So some of these are the basic information linked to the DNA. Now let's think about one of the property of DNA, which was DNA replication. Let's think about how DNA replication happens. So you are making multiple copies of DNA happening inside the cell. DNA is double stranded. So people have proposed multiple hypotheses. What are the possible ways in which DNA replication may happen? And there has been 3 hypotheses proposed: dispersive, semi-conservative and conservative. Semi-conservative has the most popular hypothesis. So let's look at this slightly in detail. If you have this DNA, let's say dark blue colored ones, both double helix. Now in the first replication after that, you can see one of the dark blue remain there and one of the new light chain, new light form has appeared. And now in the second generation, one of the dark form and the light form remains there, and again light forms again is synthesized, which makes it again double helix. This is kind of DNA semi-conservative replication. The conservative replication is based on that one DNA is always going to be like the original parental DNA, even in the after the first and the second replication. And the dispersive model says a mixture of both the strands of the DNA will be keep segregating after first replication and the second replication. So again, these are the hypotheses. Now people were kind of puzzled that what is the way of DNA replication to happen, and a group of scientists, in fact Michelson and Stahl, they did a very nice experiment and tried to prove in which way DNA replication may happen. Alright, so let's assume that we have this DNA in the parental strand, and both of them are N15 labeled. After the first generation, one of the strand from the parental one remains there, and a new form which is from the light. So this is N15. One strand comes from N15 and one newly synthesized N14. Now from this one, we have one of the blue one and one of the white, and from this again alright, and same will happen here as well, right, from this part. Okay, so now let's think about the percentage of how much we have heavy form here in the parental one for N15. So that is 100%, right? Now if I have grown N15 DNA in a medium which is light medium N14 medium, now the second strand of DNA is N14. What is the ratio here for the hybrid? This is hybrid. It is hybrid means it is having both N14 and N15 mixture of that, right? So in this particular one, in the after first replication, everything becomes hybrid, 100% hybrid forms here. Now as it goes to the second replication, how much is hybrid and how much is the N14? This is hybrid, right? This is hybrid. So these two are, as well as these two, these are part of the second replication. So what is the percentage of hybrid and percentage of N14? 50-50, right? So there are ways of doing the density centrifugation where you can separate these based on the density. They use cesium chloride based density to separate these particular bands, light or heavy form of the band, and this is how the experiment was being done. So initially they grown bacteria in N15 or the heavy isotopic forms, then they transferred that to the light form or the N14 medium, and now from there they can see the bands, is that coming in the center or there are some new band appearing which are less dense than that. And when they analyze those particular fractions, then they are able to conclude what is the ratio of N15, what is the ratio of N14 and N15, and what is the from one to next generation, how much percentage they can see the change. So if you see in this particular experiment here, in the hybrid band, which is having both 100% after the first replication, and after second replication you have 50% of the hybrid band and 50% of the light band appearing. Now if you go to the next generation, what will happen? Please draw. $25.75, somebody mentioned rightly. All right, so this is pretty much a way for them to prove that among the three models which are known, dispersive model, semi-conservative and conservative, probably the DNA replication happens using semi-conservative model, and just by growing DNA from N15 and N14 medium and analyzing the DNA contents, they were able to make this conclusion. Let me explain you this in more detail in the following animation.
According to the conservative model, the two parental strands of DNA as a whole serve as a template for the synthesis of progeny DNA molecules. Thus, one of the daughter DNA molecules is actually the parental DNA, while the other daughter DNA consists of two newly synthesized strands from fresh nucleotides. The dispersive model of DNA replication hypothesizes that the parental DNA molecule is cleaved into smaller double-stranded DNA segments which serve as a template for synthesis of new DNA strands. The segments then reassemble into completed DNA double helices with parental and daughter DNA segments interspersed. The content of parental DNA in the double helix goes on decreasing with each generation. According to the semi-conservative model of replication, each parental strand acts as a template for the synthesis of a new strand of DNA which is complementary to the parental strand. Each daughter DNA molecule always has one parental DNA strand and one newly synthesized daughter strand. Of the three replication models suggested, Messelson and Stahl proved that the semi-conservative model was correct. For this, they grew E. coli cultures for several generations in N15 containing medium, so that the bases in DNA contained N15 instead of N14. Next, they transferred and grew the cultures for several generations in an N14 containing medium. Throughout the period of growth, samples were taken, cells lysed, and the DNA analyzed by centrifugation in CSCL gradient. The parent DNA showed one band in CSCL gradient corresponding to N15 DNA. The first generation daughter molecules also showed one band which was not at the same position as parent DNA. This corresponded to N14, N15 DNA, while the second generation showed two bands, one of N14, N15, and the other of N14 light DNA. These results exactly matched the semi-conservative replication model.
So just to summarize what we learned today, we talked about two major topics. One was chromosomal basis of inheritance, and other was on molecular basis of inheritance. We learned about different experiments performed by Morgan to show the genetic basis of Mendelian's rule. We also learned about genetic recombination and linkage. And again, Morgan showed those beautifully in Drosophila experiment. We also learned about sex-linked inheritance, and we learned about chromosomal abnormalities and different diseases which are associated with that. Based on these knowledge, there are now many biochemical and genetic tests which are being used to detect various type of genetic abnormalities. In fact, at the prenatal level, at the early stage of the birth, now there are many tests, molecular tests being performed, which can detect, if there are any syndrome, any other disease or genetic carrier, the child might be possessing. And those things are possible because of our understanding of these genetic laws. And in the second part of the molecular basis of inheritance, we learned that despite not having many technology and resources, in 19th century, scientists like Griffith, Hershey-Chase, Michelson and Stahl, they performed some very beautiful experiments which has established our fundamental concepts of DNA as a transforming factor, DNA as a genetic material, and how the DNA replication happens. I hope you got good understanding of the basic genetics from Mendel to Morgan, as well as some of the molecular basis of inheritance. These are the references which are used for this lecture from your book, Campbell. And that's all for today. Thank you very much.